which will be about Facile and group diversification of polytioxazolines towards applications in nanomedicine. So for my story, I will take you to the Far East, to the lab of Professor Kazunori Kataoka in Kawasaki, Japan, at the Innovation Center of Nanomedicine. And Professor Kataoka is well known in the field for developing polymeric nanomedicine for drug delivery, utilizing PEG polypeptide blockopolymers. And my job in his lab was to develop polyoxazoline-based delivery systems for gene delivery. But as I quickly found out, in order to do this, I needed to focus on the end group diversification of the polytioxazolines to facilitate their application in nanomedicine. So actually, the application of heterotelegelic polymers in biomedicine, it's actually based on a very simple concept. You take a very heterotelegelic polymer, whereby you conjugate one end to a biomolecule or a self-assembling molecule that then triggers self-assembly, or you utilize the chain ends to attach an imaging or targeting label, or you perform reactions to play with the polymer architecture. And this concept is well established in the field, most notably conjugation of PEG or pegylation, my apologies, which, the conjugation of PEG allows prolonged circulation of nanomedicine, for instance, polymer protein conjugates. It improves the hydrophilicity, also the biocompatibility. It imparts stealth behavior, so immunogenic proteins become less immunogenic. And it's well established as an industry gold standard for biomedical applications. Polyoxazolines are another promising biocompatible polymer platform that are structurally versatile. So this R group can easily be varied to tune the structure and the properties of the polymer. For instance, you can tune the hydrophilicity, but you can also utilize this R group to introduce functionality along the polymer backbone. And you can also co-polymerize oxazolines together to arrive at polymers with different compositions, varying from random to gradient-like to block-like copolymers. So if we can develop heterotelegelic polytioxazolines, there should be ample opportunities in nanomedicine via this modular polymer platform, if we can apply orthogonal chemistry. But in order to do this, we always need to consider the end group fidelity. And when you have a synthetically diverse polymer platform, you ideally want to avoid synthetically intensive end group diversification, because then you need to think about the orthogonality of all your synthetic steps with the side chain and so on. So this is not ideal on a highly modular platform. So we want something simple and easy. So when I'm talking about the end group fidelity of polytioxazolines, then we should look at the polymerization mechanism. So typically you introduce a functionality via the initiator on the alpha end of the polymer. So you're utilizing a strong electrophile to initiate the polymerization to generate oxazolinium species. And this then propagates until you add the nucleophile to install functionality on the omega end. A well-known side reaction, however, is the beta elimination. So there is here an acidic proton, an acidic beta proton that can be eliminated and then cause a chain transfer. And then you end up with chains that have a proton as an alpha terminus. So you lose actually functionality. And this is well documented in literature. In Malditov MS spectra, I take here one of Kataoka lab. Here you can see two populations where one corresponds to the desired product and the other corresponds to the proton initiator product. Also, other groups have confirmed that this is not an artifact via HPLC methods. They have determined that this proton initiated fraction is present up to 20 mol% for polyethyl oxazoline with disperses below 1.16. But when you are analyzing these impurities, you always have to ask yourself the question whether these impurities arise from intrinsic properties of the polymerization mechanism, or do they arise from extrinsic impurities in the components of the polymerization mixture. So when I started research at Kataoka lab, I synthesized my first poly-2-oxazolene there. And I utilized standard purification protocols that I had been taught in during my PhD. And I obtained a polymer that most would consider relatively narrow, but my Japanese colleague told me that his block copolymers were more narrow than mine. So clearly there was something wrong. For a ring opening polymerization, your dispersities should be below 1.1 for sure. So I went to work, improved the purification protocols. So every component, I checked what I could purify. And indeed, I could see that I could reduce the low molecular tailing a bit, but still in MALDI-TOF MS, you could clearly see two populations, one belonging to the proton initiation, to the proton initiated species, and the other one to the desired heterotelegelic polymer. So still, I continued purifying, and then you could really see in the GPC that the low molecular weight tailing was gone, as well as a drop in the double molecular weight shoulder. And when we analyzed this polymer on MALDI-TOF MS, we could no longer observe peaks corresponding to the proton initiated product. So meaning that we have a polymer with high end group fidelity. I further demonstrated that this can also be applied to functional poly-2-oxazolines and higher molecular weight polymers. As you can see in the GPC, we have a narrow peak, narrow molecular weight distribution, and also this could be successfully ionized and presents the first example of a MALDI-TOF MS spectrum of 42 kilodalton poly-2-oxazoline. So I was quite proud of this. So with the end group fidelity, with the improved end group fidelity, we could now take a look at the end group diversification of the poly-2-oxazolines. So as I mentioned, you can initiate polymerization with electrophiles, but when we look at these electrophiles or the moieties that are introduced here, the electrophiles that you generally use, like a tosylate or a triflate, with these functionalities, they have a limited commercial availability. Furthermore, when you want to do reactions with these moieties, they have a relatively narrow substrate scope. And yeah, utilizing an azide or a cyclooctene to introduce relatively simple functionalities, this is a little bit excessive in my opinion. These functionalities are better suited for bioconjugation. And otherwise, you need to apply protecting group strategies and so on. This is in stark contrast to the omega end. So when we terminate the polymerization mixture, we add a nucleophile, which can be an acid, sodium azide, an amine, a thiol, or an hydroxide salt. So here the substrate scope is relatively broad, and all these reagents are commercially available. So the diversification is mainly synthetic exhaustive because of the alpha end group. So if you want to synthesize a heterotelegelic polymer library, then you always need to repeat the synthesis, and this is synthetically exhaustive. So we were thinking of using a pentafluorophenyl bromide or tosylate as a kind of latent electrophile. So utilizing this as an initiator, we end up with a polymer having a pentafluorophenyl group at the alpha end and an oxazolinium species on the omega end. And then the idea was to selectively add a nucleophile to terminate the polymerization reaction. And then in a subsequent post-polymerization step, we could diversify the alpha end. So when we look at the available functionalities or available nucleophiles that we can introduce via termination, carboxylic acids, amine, azide, and thiol, but also for the post-polymerization modification of the pentafluorophenyl moiety, we can also utilize the same amines, alkoxides, sodium azide, and thiols. So the substrate scope is actually quite similar, but this then poses the questions, can we apply termination conditions orthogonally to obtain these heterotelic helix? Because typically in the termination reaction, we add excess of these nucleophiles to ensure full termination. So there might be the possibility that it reacts with the pentafluorophenol moiety, as I will show later. So the first nucleophile that we investigated was sodium azide, so the azide anion. So we performed the polymerization of 2-ethyloxazolene in acetonitrile at 70 degrees. And then once it reached the targeted conversion, we added 10 equivalents of sodium azide and let it stir at room temperature for 16 hours. And then we monitored the fluorine NMR to see whether our pentafluorophenol moiety would get converted into a tetrafluorophenol azide moiety. And what we could see is at 16 hours, there was no consumption of the parafluorines indicated here next to the gray box. But after 24 hours and 48 hours, we could see signals appearing which correspond to the tetrafluorophenol azide. So it seemed at that time that we could kinetically resolve this side reaction, unwanted reaction. So once we isolated the polymer after 16 hours, we took a fluor NMR, and there we could clearly see the ortho, para, and meta fluorine signals without any trace of the tetrafluorophenol azide signals. Also, Malditov further confirmed the presence of a single population, so a single species, which corresponded to our desired product, so the desired heterotelic polymer. And also, in this fashion, we were able to synthesize heterotelic polytioxazolines in a very controlled fashion. So having done this to prove indeed that we did not form this tetrafluorophenol azide, I then proceeded to functionalize the pentafluorophenol with an azide. So incubation with 2.5 equivalents of sodium azide at 80 degrees in DMF facilitated full conversion in 2 hours, thus obtaining a bisfunctional azide. And the fluor NMR shows that the conversion is indeed successful since the parafluorines are no longer detected, and we see a clear shift in the signal of the metafluorines. And also the GPC shows that this modification proceeded in a controlled fashion, so no no polymer coupling reactions occurred. Okay, so the next nucleophile that we investigated for the nucleophilic aromatic substitution were thiols. So thiols are known to be able to react with the pentafluorophenol moiety in the presence of a catalytic base. So we screened various conditions. So we screened triethylamine, potassium terbutoxide, and DBU as a base. And it turned out that DBU was the most successful base, facilitating full conversion with merely 2 equivalents of thiol in 1 hour at room temperature. So then we obtained, as you can see from the fluor NMR, there are no parafluorines, just the signals corresponding to the ortho and metafluorines. Also, a Malditov MS, once again, we could only see the peaks corresponding to the desired product, and this was also a single population. And in the GPC, we could see a shift to lower retention times due to the introduction of these hydrophilic hydroxyl groups. And one possible side reaction that might occur during this reaction is actually the reduction of the terminal azide to the amine. But we analyzed this via cation exchange chromatography. And what we could see is that the polymers did not show any considerable retention in the cationic region. So meaning that this side reaction is not kinetically relevant under the applied conditions. Meaning that we do not get any contamination with undesired species. And we could perform this for various functional thiols. So this was compatible with hydroxyl groups, amines, acids, and we could also install protected sugars, which will probably be used in the future for targeting applications in vivo. Another nucleophile that we investigated were amines, since they are broadly available. So incubation of the polymer with amine at 70 degrees overnight in NMP yielded a clean conversion towards the tetrafluorophenyl anilines. So as you can see, we could introduce various moieties, so primary amines, secondary amines, also hydrazine could be introduced. And this reaction proceeded cleanly without any trouble. And then finally, we can also substitute this fluorine with an alkoxide. So here we used phenol as a model compound, and then we deprotonated it in situ with TBD as a base. So at room temperature, after 24 hours, we achieved full conversion. Again, fluor-NMR shows full conversion, and this is also confirmed by Malditov-MS, which shows a single population where the experimental spectrum corresponds to the simulated spectrum. So that's all good. So with sodium azide, the sodium azide terminated polymers can be easily we can easily diversify the alpha chain end of the azide terminated polymer. So now we went on to probe the selective termination with thiols and amines. But here we quickly encountered some troubles. When we add the terminator in excess, so the thiol or the amine in acetonitrile, we observed that we always get a mixture of compounds. So we get the desired heterotelegelic product, but also the bisfunctionalized product, and this is problematic. And this was actually the same for the thiol. The thiol actually proved to be more reactive towards the tetrafluorophenyl group than to the oxazoline, the aliphatic thiol, that is. So to solve this issue, we looked deeper into the mechanistics of the reaction. So when we consider the nucleophilic aromatic substitution, we know that upon the addition of the nucleophile, we form an anionic transition state, which is called a Meissenheimer complex. And typically this Meissenheimer complex, or this transition state, is stabilized by polar solvents. On the other hand, the oxazolinium species, or the oxazoline, undergoes polymerization more efficiently in apolar solvents. Due to reduced sulfatation, the activation energy is lower, so the cation is more reactive that way. So we hypothesized that if we switch from acetonitrile, a highly polar solvent, to a more apolar solvent, we might facilitate selective functionalization and obtain the desired heterotelegelics. So we did perform the polymerization then in chlorobenzene and then added our nucleophiles. And what we could see is indeed this afforded selective termination, thus maintaining the pentafluorophenyl group, as can be seen in the fluor-NMR as well as via Malditov-MS, where the simulated spectrum corresponds to the experimental spectrum. So that's all good. So with this chemistry in hand, we then wanted to explore applications for nanomedicine. And one idea, well, since I was in Kataoka lab, the idea was to synthesize ligand installed blockopolymers. So having a functionality where you can introduce targeting groups, or such as antibodies, peptides, sugars, and so on. One hydrophilic block consisting of polyoxazoline then, and then a drug loading block or a polyelectrolyte to complex RNA and so on, which would then trigger the self-assembly. So I subjected the pentafluorophenyl functional polymer to the amine functionalization with ethylene diamine, thus obtaining this primary amine and this very electron deficient amine, which cannot initiate the NCA polymerization. So this primary amine was then used to initiate the NCA polymerization of TFA protected ornithine, yielding this polymer. And this proceeded very cleanly, as can be seen from the GPC. In black is a macro initiator, and you can clearly see that there is clean chain extension with no remaining macro initiator, yielding block copolymers with dispersities below 1.1. We did explore this polymer further for gene delivery, but I will not go into detail here. I rather talk about lipid polymer conjugates. So having this functionality and being able to install thioglycerol, and then via simple Stechlich esterification, we were able to synthesize lipid polymer conjugates, as shown here. And well, we were interested in what this tetrafluorophenol moiety, which is a little bit hydrophobic, which is actually quite hydrophobic, would, if it would have an effect on the biological properties. So for this purpose, we synthesized liposomes with this epoxylated lipid and encapsulated a fluorescent dye, DID. And then these liposomes were injected in mice, and these mice were then subjected for in vivo real-time monitoring of the fluorescently labeled liposomes. What we could see for polyethyloxyethylene, it was eliminated in a two-compartment model fashion, while polymethyloxyethylene, on the other hand, was eliminated according to a one-compartment model. And actually, these half-lives that we obtained corresponded quite well to structures, so to polyoxazolene lipids, without this tetrafluorophenol moiety, indicating already that it does not have a major impact on the biological properties of the liposomes. But one of my colleagues was not yet satisfied, and therefore we explored the box-based lipids for LNP synthesis and the delivery of RNA. It was also the height of the corona pandemic, and then so this made sense. And what we know is that the polymer lipid in commercial formulation, so the DMG PEG, is actually quite crucial in not only stabilizing the LNP formulation, but also in triggering hepatic delivery. So once it is injected in the blood, this polymer lipid desorbs from the LNP and undergoes exchange with apolipo-E proteins, which then facilitate transport to the liver. And the hydrophobicity of this tail is quite crucial in this hepatic shuttling. So if the length exceeds 14 carbons, then the hepatic targeting drops and this exchange rate also drops. This has been shown in literature. So we wanted to see whether this tetrafluorophenol moiety would have a significant impact in the hepatic shuttling of the LNP. So that's why we synthesized LNPs with the C14 PEG as a positive control and C14 polyethyloxyethylene and C18 polyethyloxyethylene as a negative control. And to our delight, we show that both the PEG but also the polyoxazolene were able to provide robust gene expression or firefly luciferase expression in mice 4 hours and 24 hours post-injection, and while the C18 could not. And this is confirmed as the C18 PEG is also known to not provide hepatic targeting and thus no gene expression. So having a successful demonstration of gene expression, we then explored the possibility to utilize box-based LNPs as carriers for the COVID-19 vaccine, so for the spike protein mRNA. And we immunized mice then in a classic prime boost approach. And after 5 weeks, we then analyzed their cellular immunity and antibody production via ELISA and ELISpot. And what we could see is that our epoxylated LNPs were able to induce robust cellular immunity, as well as produce spike protein-specific antibodies. So this demonstrates that the POX-based LNPs can be utilized as a vaccine. So to conclude, this simple strategy is very powerful. So we can synthesize heterotelegenic polymer libraries very easily. And I did not show this in particular, but this approach is also orthogonal with functional poly-2-oxazolines. And there is a minimal impact of the tetrafluorophenyl moiety on the biological performance. And this is a proof of concept of the first polyoxazoline-based COVID-19 vaccine. So with that, I would like to acknowledge all the lab members from Kataoka Lab, in particular Professor Kataoka and Professor Satoshi Ushida, and also the members who collaborated on this research, as well as the funding societies, so the Japan Society for Promotion of Science and Kawasaki Institute of Industrial Promotion for financial support. And then I would like to end with a little bit of research that I do now at Leiden, which is developing polyoxazolines for biotherapeutic delivery, so continuing on delivery of mRNA, but also proteins. And here we focus on well-defined chemistry and also on purity and reproducibility. So synthesizing well-defined homopolymers, but also well-defined local polymers for the delivery of biotherapeutics. And with that, I come at the end of my presentation.